Hi, everyone. Welcome to this webinar organized at MeetUp. We are excited to start this presentation and have you as well here. We have really nice speakers that you can see some of them. If everything is all right, you should be seeing my presentation. I just do a small introduction of this webinar and what's going to happen. I'm going to give an introduction to the MasterClass, very short one, what the MasterClass is going to be about. This webinar is a meetup, a very short abstract summary of what's going to happen in the MasterClass and presentation from the companies that is going to be in the MasterClass, and also an opportunity for you to ask questions. We are recording this webinar as well, just to let you know. The masterclass will be between 26th and 28th of June, around a month from now. And for you to know, the deadline for the registration is the end of May, so next week. I would say hurry up. This is actually the second time we have the masterclass. You see a picture from last year. Jianbo is there and Katja is all the way back there, but I don't see Katja here, but she's there. We enjoyed it so much that we are repeating it another time this year around the same time. And now I'm going to give you a kind of a plan of the masterclass, what's going to happen, a bit more explanation than what you have read on the website. So first, we are going to start with a kind of a digital platform. 2 weeks before the masterclass, we send the participants access to a digital platform where they're going to see lectures, interactive lectures, quizzes and examples, and they will have access to resources and tools, and they will have access to all the materials that will be given to them through this platform during the class. Specific lectures is going to be around 1.5 hours, 2 hours. It's on demand, so they can go and see or watch the lectures anytime they want, and also all the materials anytime they want. And they will have access 1 month to this platform 1 month after the masterclass, and if they are interested, they can extend it as well. Okay, so 2 weeks before, they're going to see the lectures, etc. And then we're going to start the day with a combination of lectures and a designing program, which we call it Learn and Design. So the participants will be divided to different groups. They're going to choose an application and they're going to go through a 3-step design process based on the information they will receive and based on the feedback we will give them. First, they're going to define an application, its requirements. We call it the context of use in a team. Then they will get inspired by us, by the lecturers, and by the experts in the masterclass. And then they will use the tools we have given them, but also the chips, the systems they see during the masterclass, especially the commercial chips, they will see to design their own organ-on-a-chip assay. And this design is not a one-time thing, it's an iterative process to receive feedback from us. Again, the same day, we are going to have intestinal organoid ritual training by Stem Cell Technologies. Dr. Julie Watson from Stem Cell Technologies will give a visual training, and experts from the same company will be there to answer your questions too. Afterwards, we go to the lab. We will have a hands-on lab sessions with different types of chips, with different pumps, flow systems, and we are going to mimic seeding cells. To be clear, we are not going to use cells, but rather beads. We believe that the first interaction with the chips, you need more simpler way of interacting with it other than adding the cells. These are going to be simple academic VDMS chips, and this is a preparation for you to have a feeling how the chips work, how to connect perfusion systems to chips, and frankly, compare different chip types and perfusion systems with each other, really in practice. And a kind of preparation for the next day. Next day, we're going to have expert reviews. We have two amazing experts. One of them is here, and one of them is our co-host. So we are organizing the MasterClass in Amsterdam together with Dr. Jianbo Zhang. He's here to also show you what has been working on, a very nice aerobic anaerobic interface system to culture organoids and different cell types. And he's been improving by time his system. And I'm also curious what has been updated in the system as well. I'm not going to talk about it more because he's going to explain his work. And in addition to Jianbo, we will have Dr. Seba Withoff from University Medical Center Groningen. And he's going to also talk about organoids and organ-on-chip, specifically for small intestine, as well as in combination with microbiome. After the presentation from experts and interaction with them, we are going to have a very nice commercial session. Dynamic 42 and Mimetas will be present. You will get the chance to get hands-on experience with their system. These are really optimized commercial systems that you get to experience. And also, very important part is that you can design your idea, design your assay together with them. So if possible, if interested or interesting to you, you can work with them to design your assay based on their system. So they're going to be there to answer your questions too. And today they're going to present their own technology and what they're going to talk about during the masterclass. And then within the masterclass, so yeah, as I said, two companies will be there. And then within the masterclass, we are going to have a chips and drinks event at the end of the second day, which is really fun. You can network with different, with your peers, but also different people present in the event, companies, the host, and even more people that are going to be present. And then the last day, which is a half day, day 3, we are gonna hear your pitch presentations. First in the morning, you're gonna finalize your essay. Together with us, we are gonna be there. All the experts are going to be there to help you to finalize everything. And one important part of the process is to pitch what you have thought of. It doesn't have to be perfect. It's just has to be an initial step for your better understanding how the design process goes and how iterative it's going to be. We are going to, again, give you feedback, and this is also a very good learning process for you. Just to let you know, if in case you're not able to join this masterclass, there are the online material on the digital platform can be accessed, which I will put a link in the chat soon. And yeah, so this is the MeetUp agenda for today. I had a short introduction. Dr. Jianbo Zhang will give an introduction of his work, and afterwards we are gonna get an introduction to technologies from Dynamic 42 by Dr. Katja Graf. And then from Mimetas, Joris van der Linden will be here to show us technologies from Mimetas. And at the end, we are going to get an introduction of what's going to happen in the virtual training from stem cell technologies by Dr. Julie Watson. Great, so that was my last slide. I would like to invite Dr. Jianbo Zhang. Just before that, I forgot to mention something that I would like to ask all the presenters to disable their microphone and camera if somebody is presenting. If you have any questions, please leave them in the chat, in the Q&A section. And if you want to ask the questions, raise your hand. I'm gonna mute you and kind of let you ask the questions. And if we don't have time for questions, there is going to be plenty of time, especially for companies, if you have questions from them. And if there's no time during the presentations, we are going to have a Q&A session at the end of the webinar. Okay, great. Then Jianbo, the floor is yours.
Thank you, Hossein, for the nice introduction. Are you able to see my slide?
Yes, I can see the whole screen. Yes.
Good. I hope that this one I can minimize. Okay, good. Welcome, everyone, from my side. I'm Jianbo. I'm also a co-organizer of the workshop, and a lot of credits should go to Hossein in organizing all the events. So today I'm going to give a teaser of the systems that I have leading in building the system. And of course, this is just one of the many chips design and models that has been could be offered, including the other designs from Dynamic 42 and Mimetas and many other companies. And it's actually very good to see all different companies are entering this market, because ever since our publication and also others, I have been approached by many other potential users, and of course I cannot meet all the requirements of collaboration requests. So it's always good to see them are contributing to the field and also bring more technologies commercially available to all the users. So for this technology, we are basically aiming to design a model that is that can co-culture the primary human cells with the strict anaerobes, because we were interested in the colon. And we believe that using this model, we could understand better the molecular mechanisms, how microbes and hosts are talk to each other. And by understanding this, we can find targets or potential treatments to improve the gut health, and therefore maybe also improve their health in general, and treat the diseases that has been associated with the gut microbiome. So what we aimed to do is really focus on the colon, and it's kind of very different from the small intestine, because the conventional view of the intestine is that you have a big villi, and that is small intestine. But in the colon, it's relatively flat, or much flatter than the small intestine. Therefore, you have this kind of a layer of epithelial lining, and then you have a very deep crypts, and these stem cells are residing on the bottom of the crypt. And this flat surface is actually the most intense interaction is happening between the epithelial cells and gut bacteria. And what's difficult is our challenge is that to mimic this is that you have a very steep oxygen gradient, where in the lumen side it's nearly no oxygen, and then on the basolateral side you have a lot of oxygen, because the human cells they need it. And then a very dense bacterial cells, and also active fermentation, accumulation of toxins, metabolites, and finally the mucosal barrier with different cell types, including epithelial cells, as well as the immune cells, goblet-like cells, and many other cell types. So we aim to, let's say, meet these requirements in one in vitro model. And during this workshop, I will introduce much more details in these different versions of our system. We call it GUMI. And the first version is to build up this mucosal barrier and oxygen gradient with the microfluidic flow, so that we can culture epithelial cells, which is differentiated from the organoids, and the organoids is derived from the human tissue. And then co-culture these epithelial cells with a single strain of bacteria that is either sensitive to oxygen or relatively tolerant to oxygen. And further to that, we also tried, or this has been published just this year, that we could include different type of immune cells, in this case macrophages, dendritic cells, CD4 positive T cells, and also monocytes. So these cells are very important components in the local environment in the intestine, and being able to culture them together with the microbes you could start thinking a lot of different scientific questions using this technology. How, you know, for example, from the start of the birth, how first colonizers of microbes could train in our immune system, which has never seen any microbes before, right? And also how overwhelmingly interaction between the pathogens and the immune system could, you know, inflame the whole intestine. So this kind of questions now we can start to really answer with in this system. And furthermore, we have some pilot data that on top of the immune cells and other cell types, we can introduce a mixture of a microbial community with a defined population, so that everything in the system is defined, and you can really, let's say, tease out which microbes is contributing to what effects. So so I will stop here and leave. As I said, this is just a trailer of the workshop, and if you are interested in learning how this works and how we culture them in more detail, you're more than welcome to register and join us in the workshop. And I will hand the floor to Hossein.
Great, great. Thank you very much, Jianbo. So I think compared to last year, a new feature of your system is the immune part, right?
Yeah, and also the complex communities.
Yeah, great. And maybe we have 2 minutes, so just for the clarity for all the people here, are we going to see your system?
Yeah, so during the workshop, I will also show or have a demo of the system, so the hardware, but also maybe the transwells and how we run the experiments, and in the interface to, let's say, program the microfluidic flow.
Yeah, and you're there to answer questions and help them, help the participant with the design as well. That's already great. Okay, perfect, great. Thank you very much. Then I would like to invite Katja, Graf from Dynamic 42, and thanks for being here, Katja. And you can share your presentation and introduce everybody to Dynamic 42.
Yeah, I will do so. Hopefully technique works. Let's see.
Something is coming.
It's coming, okay.
Yes, I can see everything in full screen.
Full screen, perfect. Good, then I can start, yeah. So also a very warm welcome from my side. The next 20 minutes, I would like to introduce you into the biochips and the organ-on-chip models from Dynamic 42, with a special focus on the intestinal model and its application for microbial investigations. Yeah, my name is Katja Graf. I am the laboratory manager of Dynamic 42, and I've been working here now for more than almost 5 years. But I also got in contact with organ-on-chip models during my postdoc time, and yeah, that's there. I also studied microbial interaction and host cells, and I got to this point where I needed a more physiological model, not just a 2D cell culture, but we were interested in translocation studies. And so then I got in contact with the laboratory where also the Dynamic 42 was founded. So it's a spin-off company from the University Hospital here in Jena in Germany. And there the model or the whole technique was developed. And the company itself was established in 2018, and Dynamic 42 is not just a system provider, so meaning we offer the biochips and the whole perfusion equipment to our customers, but we are also a CRO, meaning we have our own laboratories here doing studies and try to answer the question of our customers here in our laboratory. We are then, or during these studies, we are mainly focusing on substance screenings with the help of these organ models, for instance, for different kind of drugs, but we also have experience with biological, like antibodies, or with nanomaterials that can be screened in the different organ models. But we, the last years, we also focused then on the disease modeling. So we have, for instance, an inflammatory liver model, but also quite some experience in infection models with bacterial, but also viral and fungal agents. And today I would like to focus a bit on the fungal infection of the intestinal model. You're going to show this, or I will show this then later on. The products of Dynamic 42 also are under continuous development. So we also try to, of course, improve our models and our chip systems. So at the moment, we are, for instance, very busy in sensor integration so that we can also have real-time measurements for oxygen or pH in the systems. And also the biochip itself is very flexible. So especially in terms of form, dimension, and materials, we can change the biochips to the need of our customers. On this slide, you can see here the biochip itself from a top view as well as from a side view. You can see here that it's more or less built the same way for all the different models we have. The different biochips have always a microscopy slide format, and we always have 2 culture units on 1 chip. So either for replicates, but also for individual studies. And with the help of these little ports, you can see here in the side view, we can pipette solutions into the chip systems. And then in this little window, you can see here, we have different sizes of this window. This is actually the growth area where the cells are seated in and where they can form these organ models. The chip itself is made of a biocompatible material and is also characterized by quite low drug absorption characteristics. So this is very important, especially for drug screening, so that the drug itself is not absorbing to the material, but rather stay in solution and can then be or then have effects on the cells. One example here, for instance, propiconazole is quite highly absorbed by a PDMS, a material other biochip manufacturers are using quite often. The material we are using, there you see that more than 90% are still in solution after 24 hours of perfusion. Okay, and when we now have look from a lateral angle of view inside this growth area of a chip, you can see here that the cavity itself is divided by a membrane, and shown here in gray, into an upper and a lower channel. And these are individually or can be individually filled with a suspension of cells. And then these cells settle and adhere to this little membrane and build their confluent cell layer. You can integrate also a second cell type in the same channel. And has, what you can see here is also that you can have a hanging cell layer on the lower side of the membrane so that you can build up a 3-dimensional model. The membrane itself is a porous membrane, so meaning there are pores in. This is especially important for nutrient exchange, and also other mediators can be exchanged from one cell type to the other. And there we have different materials as well as different pore sizes and densities available. Right, one step back. What I wanted to say here as well is that, of course, this rather looks like a transwell, but of course, what really makes a difference to the commonly available culture models is that this whole chip system is then also connected to a pump system. So, and this pump then sets the medium on top of the cells in motion. And this is really the difference, and also leads to differentiation of cells in this system. And this is a little video here showing this. We have a top view again. A reservoir should be here filled with a bluish medium, and you can see then in a study video that it's flushed, that the cavity is flushed. This is the culture area again for the cells, and then this looks like this. So medium is flushing the cells, and you can see here the medium is underflow, and this can be done in both channels, the upper and the lower channels individually. Okay. Next slide. This is how it looks then inside the incubator. So that it's actually all you need is an incubator. Then the pump itself is also standing inside the incubator. The chip, and here you see now in this little petri dish, is also inside the incubator, and you can plug on this little chip different kind of reservoirs. We have little and bigger ones for different volumes of medium, and together with a tubing system, then we can connect this to the pump, which then leads to an sterile closed circular system. So the medium is always going from the reservoir through the chip, through the tubing, coming back to the reservoir, so that we really have a closed system, which is also necessary if you want to have accumulation of certain mediators like cytokines or whatever you like to measure in the system. This is pretty new. I just put it in to let you know we will launch a starter kit very soon. With this starter kit, you can then able to work, or you can start immediately in your lab. The only thing you need is an incubator. So you get the chips, a pump, and all the perfusion system. So then, yeah, we are pretty well equipped to start. Okay, let's come to the biological part. The organ models we have available in Dynamic 42 are the 3 ones shown here on the slide. We have a well-characterized liver model, the intestinal model, as well as a lung model. And what they all have in common is that we are integrating a vascular compartment in one channel, for instance, on top of the membrane or also below. That does, depending on the model you're using. So this means we are integrating organ-specific endothelial cells, sometimes always also mixed with immune cells like resident macrophages. You also have the possibility to go for perfused immune cells within the vascular compartment. And together with the lower channel, then where we are integrating epithelial cells of the organ, this builds, let's say, the 3-dimensional model, which is more or less the smallest functional unit of the organ you can build. And this is, as I said, the 3 models are well established, but the chip itself, of course, can be also used for other models, and you can integrate other cells, other organs can be built up that answer your specific scientific question. What you can also do within the chip, having, for instance, on one cavity built up in the intestinal model, on the other side, for instance, a gut, a liver model, so that you can also have here multiple organ connections. This is something we use in our labs for ADME studies, but as well as for inflammatory processes or infection models. Don't know where this video is shown here. Doesn't matter. The readout options you have are more or less the same you all know from conventional 2D models. So with the biochips from Dynamic 42, you can have supernatant sampling. You can do everything you like with the supernatants, like measuring clinical parameters, cytokine profiling, compound turnover when you have analytics in-house. But we also use the supernatants for barrier assays, so measuring, for instance, fluorescently labeled tracers that are pivoted in one channel, and after a certain time frame, we measure what comes to the other side of the tissue, so that you see the barrier integrity, is it okay or if it's not okay. You can also extract the tissue from the chip, meaning that we can cut it open and it can get this whole organ tissue out of the chip, and then you are free to choose your staining method. You can use it for IF staining or for electron microscopy, whatever you like. But you can also do single cell suspensions then again from these cells for flow cytometer, or you can lyse the cells for RNA or Western blotting or whatever kind of material you like to extract. I told you already it's a microscopic slide, so you can put it also in any kind of microscope. And if this is suitable for live-cell imaging, this is something you can also do, and I hope this works now. What you can see here are fluorescently labeled PBMCs on top of an endothelial cell layer. And after a certain stimuli, the cells started to attach to the surface, and then they migrated into the tissue. So this is also something you can do directly inside the chip. Therefore, you don't need to cut it open. And last but not least, any kind of enzymatic activity assay is also possible to perform it inside a chip. Just an example, we, for instance, in the liver model regularly measured the CYP3A4 activity by simply pipetting a substrate inside the chip and then doing the normal assay procedure. Okay. So then I would like to come to the intestine model. Our intestinal model is built up as shown here in this little figure. So we have, as I mentioned before, a vascular compartment. You have the porous membrane and an intestinal compartment. And for the vascular compartment, we usually use HUVECs as in the endothelial cells. These are mixed then with monocyte-derived macrophages to get the model immunocompetent. And then we have a hanging cell layer with CACO2 cells. And what is really always very fascinating for me is to see how the CACO2 can develop if they get the right stimulus, meaning what you can see here also on the side, if you just cultivate them statically, also inside the chip, they form a monolayer with little hills, but maybe not really a three-dimensional structure. But if you perfuse them just for 7 days, and doesn't need longer. So also the differentiation in static usually takes 2 to 3 weeks. We already see after a few days what these cells are capable of. So they really start to form villus-like structures. They have a huge outgrowth of cells. And this is also shown on the next 2 slides. Here again, in the cross-section, you can see static versus perfused cultures. So the monolayer and the static, but you have here 100 micrometer thick microtissue when you perfuse these cells. So this shear stress they experience really lead to an outgrowth of villus and crypt-like structures. And this is then also reflected by a drastically reduced permeation of FITSI-labeled tracer dextran. So the barrier functionality really increases upon this very thick tissue. And the slide here again should show you what I've just said. So this is a top view of the cells that has been taken with just normal cell culture microscope. And we start now at the villus tips and then go in that direction towards the membrane. And this little video should give you again an impression of how extensive the 3D structure is in the gut model. So you can see here the villus tips coming. And then we go further inside the tissue to the basolateral side. Okay. We characterize this model by having a look at certain markers that are expressed by the cells. You can see here for the vascular side that endothelial cells are staying positive for CD31 as well as for VE-caterin, and that we also have the macrophages present that are CD206 positive cells, seen here in green staining. And also the intestinal cells are stained, the enterocytes, for instance, for adherence and tight junction markers like E-caterin or SETA1, and villine also expressed then at this villus tips here is also very prominent and very good visible. And besides the enterocytes, the CACO2s can also differentiate in other cell types of the intestinal layer. So for instance, Paneth cells are present as well as mucus secreting goblet cells. Here, for instance, shown that the green cells here are MUC5IC positive cells. So we really have a differentiation of different cell populations under dynamic perfusion conditions. What we do with these models is we use them for different kind of studies. Of course, we can have toxicology testings, you have nutritional studies, drug delivery or absorption of drugs to the intestinal compartment can be studied. And today I would like to show you two different or two models or studies we were performing in our labs. One is a disease model for IBD, so for inflammatory bowel disease, but also an infection study with Candida albicans. Good, starting with the first one, I said already, inflammatory bowel disease is something many researchers, of course, interested to find novel therapy options. And I think most of you might be aware that IBD patients are suffering from a disturbed intestinal barrier. So they have the paracellular permeability is increased. This leads then to that bacterial proteins and so on can pass the barrier and reaches the vasculature. This leads to infiltration of immune cells and inflammatory processes there. And this is something what we can also mimic in our chips by a special chemical treatment. And this treatment leads to this IBD phenotype, meaning we have disturbed junctions in the intestinal and vascular compartment, shown here again in IF staining for E-caterin and V-caterin. This is then also reflected by an increased permeability for these physiological traces, shown here in this graph, the control versus the IBD treatment, leading into an increase. This also is reflected by reduced villus height compared to the control chips. But what was also quite good to see that this is really something that affects the barrier, but not the viability itself. This was also expected and could be shown here in this test. Yeah, and what to do now with these chips? Again, here's some IF pictures showing E-caterin in green, and under IBD conditions also in red immune cells that are infiltrating into this tissue. Yeah, with this model, of course, we can test novel therapy options. And this is also something we tried. We did this by integrating not only the chemical for the IBD conditions, but also having the presence or the coexistence of a probioticum. Here we used Lactobacillus rhamnosus. This together with the IBD stimulus was able to rescue this IBD phenotype, and rescue rather preventing it. So you can see here then when staining the junctions again that they remain intact when this probioticum was present. This was quite nice to see. And this was something, of course, we could continue in many other scientific studies of how this works. And yeah, there are many options for further experiments now. The second application I mentioned already is an infection model. This was in cooperation with the Hans Knoll Institute here at the Beutenberg campus in Jena. They are really experts on fungal infection models. I also did my PhD and my postdoc time there, and I was working with Candida albicans and the infection of intestinal cells. And in this study, this was the first time where we also used a microphysiological model, because here we were able to have not just having the fungal cells and the treatment on the same side of the cells, but really can mimic an intravenous administration of an antifungal drug. So we did so by applying this drug in the vascular compartment, and we wanted to see if this drug has an effect on not only on the Candida albicans wild type strain, but also on a clinical isolate. And shown here is just that you can see that the presence of Candida albicans on a normal looking tissue in the control chips really leads to the formation of hyphae and microcolonies that is disturbing the tissue and is also decreasing the barrier. And by applying an antifungal drug, in this case caspofungin, we could see that the wild type strain, this is again here the growth area of the chip, is indeed affected by a drug that was administered in the vascular compartment. So you can see here that microcolonies on the tissue are decreasing in a dose-dependent manner, whereas the clinical isolate we were using as a comparison, this was an echinocandin-resistant clinical isolate, was not affected at all. So this means, yeah, you can, or it proves that an intravenous administration of a drug is possible, so that the drug can pass the cell layers and can reach the fungus on the other side and has an effect there if it's responsive to the drug. And again, we used our lactobacillus strain as a probioticum again to see if this is also a treatment option for Candida albicans infections. And here, the bacterium as well as the fungus strain were applied both in the lumen and the intestinal compartment. And we were also able to see that the bacterium had a positive effect on for the host tissue, but not for the fungus. So the translocation of cells were drastically reduced, of fungal cells, of course, in the presence of Lactobacillus rhamnosus, and this also leads then to an reduced fungal burden and reduced host damage, which was measured by LDH and stupinatants. So there we had a significant effect if the bacterium was present. Okay, this was a little excursus in the gut model and the different applications. I would like to summarize very quickly, what the advantages of our organ model or organ on chip platform is. So I said already that we have a standardized chip that we can offer our customers. So it's a microscopic slide format, and also the ports we use for pipetting have a standardized lure format, so that the perfusion equipment you can use, you're free to enjoy what kind of systems you would like to use. You don't need any proprietary equipment. You just need an incubator, and with help of our starter kit, you can start immediately. Every chip you purchase as or needs to pass a quality control. So we are certified by an ISO 9001 system here in Germany. And yeah, what also our customers told us is that it's really not witchcraft at all. It's something you can learn. The handling of the chips is not that difficult. Sorry. And yeah, I think an experienced scientist can start immediately. And this is actually also the things we will show you then in the masterclass, is that how to use or how to pipette a solution into the chips. That you can start with a cell suspension. We don't use the cells, we just, therefore we don't, we have not the possibility to cultivate in advance, but the pipetting itself, we will practice. We will also practice how to connect these pumps to the perfusion system, to the pump, how this is all then started, that the circular system is closed and the perfusion is set up. And we will also practice how to extract the tissue from the chip, how to cut it open, and what you can do then afterwards. So then I think the first steps then are done, that you are able to start to work. With this, I'm at the end, and I would like to hand over to Hossein again. Oh, I of course, welcome to answer your questions.
Yeah.
Yeah.
Thank you very
you
much,
can answer the question.
I wouldn't dare. No, great. Thank you very much for your presentation. Very nice presentation. There is one question. I will let Jianbo ask that. But before that, I would like to praise two things, because many times we forget about the practical part when we want to choose an organ chip model. So two things I want to praise is that first, you kind of established drug absorption in your model. Very transparent that there is low absorption in your model, which you do not really see often in organ and chip models. The next one is the really good thing is that you can cut the sample out, which also you don't see often in other models. It's often dissociation of the cells from the chip, or kind of lysating all the cells out of the chip, but you can cut it directly from the chip. So I really like that. So I will like to ask Jianbo to ask the first questions. There's a question in the chat.
Yeah, so Lena Neufeld has a question. How many repeats you can run to connect this system?
How many with one chip? Do I get that correctly? So the chip itself is a consumable. So unfortunately, at the end of the experiments, so when you cut it open and extracted the tissue, it's of course not reusable anymore. So all the perfusion equipment, the reservoirs, the tubings, the pumps, of course, are reusable, but the chip itself is a consumable. I hope this answers the question. Yeah.
And maybe also out of my own curiosity, how many, let's say you have one chip that is running, how many chips you can run in parallel? Maybe many of audience are also interested in that.
Yeah, of course, a fair question. I have to admit here, it's definitely not a high throughput method. This is really also different to the talk afterwards, I think. It really it's not. So we have the pumps inside the incubator, and of course pumps are producing heat, so we can't put that many inside. And depending on the model, we have 3 pumps in. At one pump you can fit 4 chips. No, I'm wrong. 2 chips, of course, because you have 2 cavities per chip. And if you perfuse one side, what we actually do, for instance, for the liver model, then you can plug in 2 chips on one pump, meaning with 3 pumps in an incubator, you have 6 chips running in one incubator. So that means really that you need to have more than one incubator available for the system. If you want to have, let's say, 6 to 8 or even 12 samples in parallel, you would like to address, then you need more than one incubator and also more than one pump.
That also means you can roughly have like 6 replicas in running and 3 and 3 for each conditions if people want to do experiments.
Yeah, this is possible. Yeah.
Cool. Thanks.
Yeah, so I have 2 questions. We still have time. So maybe a bit about the size of the membrane and the pores that you have in your chips.
So starting with the pores, we have different pore sizes available. The biggest is 8 micrometer. It goes down to 3 micrometer, but also 0.4 micrometer possible. I'm aware that different kind of cells need different conditions. So for instance, lung and primary cells, they rather prefer small pore sizes. Others, they don't care at all. So, but they're we are free. We can also integrate different materials and different that have different processes. So if a certain type of membrane is available on the market, we can give it a chance to integrate into the system. And from the size, so from our standard chip, we have approximately the size of a 24 well plate, so almost 2 square centimeters. But as seen also in one of the slides, we have smaller chips where the growth area is much, much smaller, especially for precious cells. So if you have IPC-derived cells or very expensive primary cells, you don't want to waste that many cells, you can go for the smaller channels for the smaller growth area, also possibility.
Yeah, great. And a follow-up on that a bit. So I know that using organoids or primary cells is quite challenging. Do you have some experience with that as well?
We do have primary cells in the liver and the lung model. In the lung model, we are establishing at the moment, or it's almost done, an alveolus as well as a bronchial model. This is working pretty well. It's always depending on the donor, I can tell. So especially in the bronchial model, until we reach this phase where you have these ciliated cells under air-liquid interface, it takes, depending on the donor, 2 or even 5 weeks of differentiation, so something in between. But this works, and they also look pretty cool when they start to have these ciliated cells then. And primary hepatocytes, we also have a lot of experiments. This is working. In the gut model, unfortunately, it's not so easy. So working with organoids from adult stem cells, I think you're aware that there's a patent on this. So we, unfortunately, here in the company are not able to work with this. And we have started now testing also primary epithelial cells but this is still under development, so I can't tell that much about this at the moment. Yeah.
Great. Thanks.
Right, because of there's still 2 questions, but because of the timing, maybe Katja, you could answer that in the chat.
I can try. Yeah.
Great. Thank you.
You're welcome.
Great, thank you very much, Katja. There are interesting questions also in the chat. So maybe if we have time at the end, after you answer them in the chat, we can also discuss them in detail at the end of the presentations about cockatoo cells. Okay, great. Thank you very much, Katja. Now I would like to ask or invite Joris van der Linden from Mimetas. Hi, Joris. Nice to see you. Hi. To share your presentation, please, and we look forward to seeing your presentation.
Of course. All right. Let's maximize. All right. Hello, everyone. My name is Joris. I'm a field application specialist for the EU and Asia at Mimetas. I started last year in July, so I'm almost a year at the company now. And well, before that, I did my PhD in a completely different subject. So since a year ago, I've been pleasantly surprised by the whole organ on a chip world. And hopefully I can explain a bit more about that today to you as well. So today I would like to talk about 3D intestinal models in the organoplate. The organoplate is well the main system that we have, and I want to introduce that today, and I also want to show some examples of the applications that you can do with it. But first of all, I want to give a bit of an introduction in the goal of Mimetas, the aim of Mimetas. What do we want to do? So the mission of Mimetas starts with the fact that we all get ill, basically. Many of these diseases we can cure by now, but there are also quite some that we don't have a good treatment for yet. Think about ALS, chronic kidney disease, and also IBD, of course. The models that we have so far in the labs, like simple cell cultures and animal models, have proven to be very bad in predicting whether or not a drug is going to make it through clinical trials. We've seen that drugs that end up in clinical trials often fail. Well, and of course, then all of the research that went into that drug is basically lost time and lost money. So what we want to do at Mimetas is we want to find new human disease models in the organ on a chip platform that we have, the organoplate. And how we want to do that is by really taking human disease models, like organ niche models, into our organ on a into our chip model, basically. These models we can make as simple as we want to really understand, I don't know, the fundamentals of these diseases, but also as complex as we want. And that's what we aim to do. And in the end, we would like to find, well, novel therapies, of course. So how do we do that? We do that using our organoplate family, consisting of 4 different plate types. Each of them their own advantages and disadvantages. The first plate type is the organoplate Tulane 96, which has 96 chips in one plate. Each of these chips consists of 2 different lanes, as you can see here. One lane is the ECM channel, in which you can put an ECM gel, and the other channel is a perfusion channel, in which you can put any liquid you want, any cells you want, etc. I don't want to focus on this plate type today, because for the intestinal models, the next 2 plate types are way better. So I will continue to explain the 3-lane 40 and the 3-lane 64 plates, which are quite similar to each other. They behave very similar. They are just a bit different in the layout. And the 3-lane 40 has 40 chips in one plate, and the 3-lane 64 has 64 chips on one plate. Both of these chips have 3 channels. Again, an ECM channel in the middle, and then 2 perfusion channels on the sides. Here I want to highlight also the phase guides that are, well, not really separating the 3 channels, because the channels are open and liquid is able to flow through. However, these phase guides are able to contain the ECM in the middle channel after you put it into the channel. And you can nicely see that here. So the ECM, after you put it in, it stays in the channel and makes this curved shape towards the sides. And then after the ECM became a gel, you can add liquids into the side channels and seed cells. More about that later. First, I want to also mention the organoplate graft, which is a bit more complex, but it allows for more complex models as well. It looks similar to the 3-lane 64, with the difference. The main difference is the middle channel. The middle channel is wider, allowing for more in-gel culturing of cells. And besides that, there's also a hole on top, which allows for direct access to the ECM. With that access, you can, for example, add a spheroid on top, or you can culture a monolayer of cells, which also allows you to create an air-liquid interface for a lung model, for example, or skin models. All right, so today I want to talk about the 3-lane 40 and the 3-lane 64. And the most simple model that you can make here is this model, basically. Again, there's the ECM gel in the middle, which makes this nice curved shape, against which you can seed cells, basically. If you then start perfusing by putting it on a rocker, we use a rocker to have bidirectional perfusion. We don't have the pump systems. Upon perfusion, these cells will nicely grow out and form a very nice 3D tubule. With that, you can, well, do your main assays, and that's already very, very cool to use. Besides that, you can make it more complex. For example, you can add cells in gel. You can add an extra tubule at the opposite lane. And you can also make a co-culture of different cell types within the tubule. All right, so what you then get is a model that looks like this. At Mimetas, we've established quite some models by now, including epithelia, connective tissue, vasculature, and immune system. And all of these models you can use for robust assays. So a very strong point of our plates, because it has a very thin glass bottom, you can easily use it for microscopy, but you can also use it for biochemistry, ELISA, RNA sequencing, etc. Because our plates are in a 384 well plate format, it's also compatible to put it in high throughput imagers, plate readers, liquid handlers to automate some of the workflows. And with that, you can really scale it up to do large screenings if you want to. So how do we work together with customers? First of all, partnerships with mainly pharma companies, where we work side by side, really to establish a complex model in order to develop drugs. We also offer services. So if customers want to outsource some of their projects, we can do it in our own lab here in the Netherlands. But today I mostly want to focus on the products that we offer. So you can build your own model in your own lab. If you choose to do that, you will get the guidance of me. So I will help you set up the model, and I'm available for any technical support. So I'm not going to talk about the partnerships and the services today, but more about how to do it on your own. So you will need organoplates for that. You will need equipment. So the organoflow, that's the rocker that induces the flow through the channels. And we also have the organotier, which I will explain later, which is a nice addition to the whole model. It's not necessary to have. We also have the organoready system, which are our plates, basically our 3-lane plates, with pre-grown 3D models in it already. So we already loaded the collagen, and we loaded the cells in the chip. And these plates we can ship to customers, and thereafter a bit of recovery, they are ready to use in the lab for any assays you want. So today I want to highlight 3 of these organoready products. First of all, the organoready collagen 1, which already has the collagen 1 ECM in the middle, and then you can seed your own cells in the side channels, and you can choose your own cell types, of course. We also have the organoready colon Keiko2, in which we already seeded Keiko2 tubule, and we have the colon organoid organoreadies. Both of these I will show in this presentation as well. Starting with the Keiko2 organoready. And for that, I want to highlight our latest publication of this year, in which one of our scientists, Moran, he used the colon Keiko2 model to look at the Keiko2 tubule response to enterotoxins. So upon treatment with enterotoxins, he wanted to look at several readouts, so several ways to see what's happening there. First of all, he wanted to look at barrier permeability, for which he used the organotier that we have. Then we used, he looked at cell toxicity using the DRAG-Q7 staining, but also using the LDH assay. And then he wanted to look at the morphology of the tubule and the cells, the individual cells, using microscopy. So first of all, the organotier, it's a very small device in which you can place your plate, and then upon assembly of the whole device, these very tiny electrodes are sticking into the wells, which are voltage sensing electrodes and current carrying electrodes. And with that, with the voltage and the current, you can determine the resistance across the cell barrier. So the resistance of the Keiko2 cell barrier is a measurement for how good the barrier function is. High resistance is a very good barrier. Low resistance is a very leaky barrier. So then he exposed these Keiko2 tubules with several enterotoxins, nigericin, ochratoxin, patulin, and melittin. And all of these, you see a very nice drop in tier. So a very nice, drop in resistance when you increase the concentration of these toxins. You see that in each of these ones. The way we can do all these concentrations and multiple multiple toxins is because we have 64 chips in one plate, and you can, well, make multiple plates at the same time. So you can really do a lot of replicates as well. This also allows to determine the EC50 of each of these toxins. Right, then it's the question, well, how does this work? How do these toxins lead to a disruption of the barrier? First of all, Moran looked at the toxicity of the toxins, and only 2 of them he saw an effect, a toxicity effect. So when he looked at the drug Q7 staining, he saw that ochratoxin has a very, very minor cytotoxicity, whereas the melittin has a very nice increase in toxicity when you increase the concentration. However, the other 2 toxins, they didn't show anything. So something else is going on there. And for that, he looked at the morphology of both the tubule itself and the individual cells. So first of all, the tubule itself, nigericin, patulin, and ochratoxin, you don't see a lot happening. However, for the melittin, you see a big disruption of the barrier, which is in line, of course, with the data I showed on the previous slide. When we look at the actin staining, we can really see the effect, the morphology of the single cells. And in the case of nigericin, we saw that there are these clumps of actin appearing, which you can see here in the form of puncta, which you don't really see in the vehicle-treated cells. This is also nicely shown in the graph right here. And this nicely shows that nigericin, although it's not cytotoxic, it causes barrier disruption in a different way. All right, so I just showed you how to use colon CACO2 models for the evaluation of different modes of action by enterotoxins, either cytotoxic or morphology. And I showed that this is high throughput. However, I showed a very simple model, so just the CACO2. But it's also possible to add some complexity. So you can add both active or inactive bacteria to the tubule. You can add immune cells, cytokines, and multiple cell types in a tubule, like we did here in this IBD model, where we co-cultured CACO2 with the mucus-producing HC29 cells, and we added all the immune components to it. So that's something you can think of when going for more complexity. Speaking of multiple cell type tubule, we also have a different organoready product, the colon organoid product, where compared to other cell types and products, our colon organoid organoready product is proliferative, it is differentiated, and it has different cell types, which I will show you in a bit. And one of the big advantages is that it's license free. So if you buy it from us, you don't need to have a license. So how do we make these colon organoids? We use adult stem cells. And then we use hub organoid technology to expand these adult stem cells. We then put these cells in our plate, and we have a very nice polarized, luminized tubule. So what does that look like under the microscope? So first of all, there's the organoid expansion. Then there's the tubule. And if we look at, this little piece, the side of the tubule, we see that there is very nice polarization of the azarin marker on the apical side. Besides that, there's also the tight junctions that we want, of course. And here's another staining of Esrin and Villin as well. So we've shown that we have enterocytes here and that there is polarization, but we also wanted to see if there are other cell types as well. So for that we looked at the tubule, and we took first the bottom of the tubule and made max intensity projection, and we also took the top of the tubule and did the same. So when we looked at the MUC2 staining, we saw that there are nicely mucus-producing cells apparent in the tubule, both in the bottom of the tube and the top of the tube. Then we also looked at Ki67 staining, which is a marker for proliferative cells, and we also see that these are there both in the bottom and the top of the tube. So this nicely shows that there's a consistent cryoarchitecture. We have the goblet cells producing mucus, and we have the proliferative cells as well. Then there is a little bit of time left, so I will go over this quite quickly. So what can you do with this? We used the rhodamin to study transport from one side to the other side. And rhodamin uses the PGP transporter to be transported across the cell layer. So what we did here in this experiment, we added rhodamin to the basolateral side and the apical side, and we see that from basolateral to apical we see a very nice transport. However, from apical to basolateral we don't see any transport, which shows that that the cell layer is indeed very nicely polarized. When we then block the PGP transporter, then we don't see any transport also from basolateral to apical. So this is just a quick example of what you can do with these colon organoids. Then I would like to end. I hope that I've shown you that that you can use our intestinal models from ranging from a very simple colon KCO2 model to a complex co-culture model to the organoid model. And that you can use that with robust assays such as microscopy. And hopefully this will have a positive impact on the therapy development. So with that, I would like to thank you all for your attention, and if there are any questions, please feel free to ask.
Great. Thank you very much, Joris, for the nice presentation and the overview of the technology you had. I have a few questions. I would like to ask Jianbo also to, if he has questions, bring up questions. Maybe I start. I don't see any questions in the chat yet for Joris, but I think we do have enough. First of all, I actually really like personally Organoid here. You know, in one go, you can get a lot of information from so many chips. Also, in these two presentations, we saw different types of technologies, different complexities, different throughputs. That's actually really nice overview to have. My first question would be whenever you add a treatment, drug or toxin to the system, how do you add it for the understanding of the audience, and how long does it take until you see an effect and the molecules reach the tissue model?
You basically just replace the media in the wells. So you take out the media without the toxins or the drug, and you just add the media with the drug, and well, it directly, because of the flow on the rocker, it directly reaches the right tissue. And then it completely depends on the drug or the concentration, how soon you will see an effect, of course.
Right. And how much is the volume in the reservoirs compared to the inside the chip, inside the channels?
Inside the channels, it's about 2 microliters, and in the reservoirs, it's 50 microliters, 50 per well. So
Per well, yeah.
in one channel, you have 2 reservoirs, the inlet and the outlet, and in both, you have 50 microliters.
Right, right. So the treatment really reaches there very fast and compared to the media that's already in the chip. Okay, that's clear. And can I ask the next question, Jianbo, or do you have a question? Yeah, okay, I'll go on. So in the context of barrier disruption, right? You have organotier, you also showed, I think, FITC, kind of a dye. So I would like to know what's the difference in respect to sensitivity, and do you also see correlation between the tier measurements you have and the FITC measurements?
Yeah, I don't have a slide here about that, but we do see correlation, and we see that the organotier is more sensitive. So effects that we don't see yet in the FITC dextron assay, we already see effects in the organotier. Besides that, it's a bit more work to add the dyes to each of the wells compared to just putting all the plate in the organotier.
Yeah, yeah, that's clear. And of course, you use microscopy to kind of detect FITC diffusion to the gel. Do you have automation protocol for the image processing, or can you help the customers with that?
Yes, we can help to some extent. So we in-house use the molecular devices, high throughput imagers, so the HT.ai system as well. So we can offer there a bit more support because we have hands-on experience. But yeah, so it's a 384 well plate format, so you can just in these high throughput imagers you can put the plate dimensions and all that stuff. So that allows it for high throughput imaging as well.
Great. Yeah. Thank you very much, Jianbo.
Yeah, if I step into users' shoes, that I was curious that the volumes in your organelle plates seems to be very, very low. And are you able to recover enough volume to do a lot of different analysis? And also, how do you handle the foam forming? Because the medium also contains a lot of maybe proteins, and how do you handle that practically? Would you show us during the workshop as well?
Well, the foam formation, we don't really see actually, so we don't have a lot of foam. With regards to the other question for the assays, we do have to pull multiple chips because indeed the volume is very low. The number of cells is very low that we get out of the chip, so you always have to pull a number of chips depending on what you want to do.
Thanks.
Great. Thank you very much, Joris, to be here, and we look forward to having you in the MasterClass.
Me too.
Great, thanks. I know that there was a question about membranes and about the production of the membranes from Dale. So in general, many of these membranes are produced commercially and then integrated in the chips. There are usually drag-edged membranes. Let's say in the case of a few companies, there are PDMS membranes as well. In the masterclass, we do not focus on the fabrication of the chips. We have an overview of different fabrication methods in our lectures, and we compare them to each other. But we mostly focus on the application, different technologies available in academia and specifically commercially, and try to help you understand the strengths and weaknesses of each chip model, let's say. But we are there to answer any questions. So if you have questions about fabrication, we can go through the fabrication process, show you how things work. And one important thing is that after the masterclass, we're still there to support you. So we can have personalized meetings with you regarding organ-on-chip to help you with your specific case. So just to let you know. I have no addition, Jianbo. Do you?
No, just to leave the time also, maybe if there's still burning questions, you can still maybe raise your hand and let Hossein know. And if not, yeah, we can close the seminar.
I think so. I think so too. They can always feel free to send us an email if you have any questions about the masterclass. Again, the deadline is next week, 31 May. And hopefully we can see you in the masterclass. Thank you all. Thank you, the speakers, Julie, Katja, and Joris, and as well as Jianbo. And hope to see you soon.
Thank you, and thank everyone.